Okay, so it's my great pleasure to chair the next session, which is focused on the Accelerating Novel Therapies theme. So we'll start with a short video introducing this theme.
Of the estimated 6,000 rare diseases, 3/4 affect children. The majority have no effective treatments, and 1 in 3 of these children will die before their 5th birthday. Those who do survive will often have a very different life to their peers. We hope to change and speed up the pipeline of delivering new treatments for children with rare diseases and the adults that they will become. For example, since 2017, we have helped to deliver over 130 phase 1 and phase 2 clinical trials, from RNA therapies to enzyme replacement and cancer treatments. In the same time, our work has contributed to regulatory approval in the USA and Europe for 11 new treatments for childhood rare conditions. These are now standard treatments outside of research. Hundreds of patients at GOSH have already benefited directly, and many more will have their lives changed from this work beyond our walls. This is the legacy that we must continue. We plan to focus on 3 main areas: developing models of disease for discovering new treatments and biomarkers, creating a pipeline that allows us to test new treatments before clinical trials, and finally accelerating clinical trials themselves through nurturing our close links with industry. Our work will have far-reaching consequences within children's medicine, improving geographic access to life-changing and life-saving treatments within the NHS and far beyond, for the 17 million people worldwide who have a rare condition.
Okay, so it's my great pleasure to introduce Professor Paul Gisson, Professor of Paediatric Metabolic Medicine and an honorary consultant at Great Ormond Street Hospital. He leads the Gene and Stem Cell Therapies theme at the BRC, and he's going to talk to us about some groundbreaking pioneering work in Batten's disease.
Thank you very much. Thank you very much, Manju. Hopefully you can hear me. Hello. Okay, so I'm Paul Gisson, I work here, and first of all, I'll hit you with what I'm going to talk to you about. So over the past 10 years, our team, and I do represent a large team here at GOSH as well as ICH, so we contribute to development of the first ever successful and effective treatment for Batten disease, which is an awful neurodegenerative disorder. And that trial that started from a phase 1 trial then went on to the pivotal trial and led to the registration of the enzyme replacement therapy in this disorder, both FDA and EMA, and then eventually approval by NICE for reimbursement. We managed to transition this essentially a clinical trial situation from the clinical trials unit onto the NHS wards, where first ever nursing team managed to actually access and deliver these treatments on the NHS ward, which was unbelievable for the generally for the medical community around the world. We led the NICE approved managed access agreement to deliver this drug on the NHS, and during the COVID times, I would say heroic team led by the nurses managed to deliver this drug to all the patients in the NHS, English NHS, and also in the Northern Ireland. Then we supported development of the 5 new centres in the NHS England and also 1 in the Northern Ireland, and transitioned patients for the delivery in the local, in their local centres. In addition to that, also during the COVID times, we delivered first in the world enzyme replacement therapy administered in the eye, and also with our wonderful biomarker discovery group, supported by the BRC, developed a biomarker of treatment in this condition. So first of all, what's CLN2? It's a disorder, autosomal recessive disorder due to the biallelic mutations in the CLN2 gene, which encodes a lysosomal enzyme. The estimated incidence is about 1 in 200,000. It's a bit more common actually in the UK. And what happens is these children develop relatively normally. Most of them are fairly normally until the age of 3, and then they start losing their mobility, their language, they develop epilepsy, and then they lose sight and become blind as well. That's the natural history. The life expectancy in natural history studies is about 10. Some patients die earlier, some patients just a bit longer. The treatment, as I told you already, the new treatment that we helped to develop is the enzyme replacement therapy, which is delivered through the ICV device I'll show in a minute, and a lot of it until now was supportive. I'll show you this neurodegenerative scale because just to make so that next few slides make sense as well. So as I said, this is a neurodegenerative scale developed in the patients who had, in the natural history study, who did not have the treatment. Essentially, there are 2 domains in that scale, language and the motor domain. 3 is the top mark that they can get when they have a relatively normal, age-appropriate function of their language and motor. And then as they have minor loss of their function, they go down to 2. And then when they have major loss, get down to 1. And then when they lose their function completely, then they go down to 0. So the maximum points they can get is 6, minimum is 0. So here you see what actually happens in the natural history. So these patients, as I said, develop fairly normally, and at the age of 3, they're still, most of them will have 6 on combined motor and language scale, and then they start rapidly decline. And by the age of about 5, they're down to major loss in both language and motor. And that's unfortunately when these patients are diagnosed. So there is a delay in diagnosis. And in the MRI scans that you could see on the side, it's the increase of the white that you could see on the picture, and that means that they lose their gray matter. So they lose their cerebral function as well as volume. So this is the cerliponase alpha, or also known Brineura, is the drug, the enzyme replacement therapy that has been developed and now approved for this condition. It's administered through this intracerebral ventricular reservoir that has to be put in by the neurosurgeons, where it sits there, and then they come in for infusions every 2 weeks, and that infusion goes on for about 4 hours. So this is the summary of the result from the clinical trial. What you see on the graph is essentially 0 is their baseline points on that CLM2 scale. And then as they lose, because they can only lose certainly points, certainly in the natural history study, as they lose with time, they lose these points. So what you see here is that in red, that's a natural history controls patients. Over about 4 years, they lose 4 points because at the beginning they had already lost some, so they probably had a maximum of 4 to start with. Whereas the patients on the enzyme, in the first 6 months or so, some of them lose some of the points, and then after that they maintain their function for about, certainly for 4 years afterwards. So, as I said, in order for this to happen on the wards in the NHS hospitals, the best way we saw this is for the clinical nursing teams to take on the administration. This is the safest way to administer drugs, rather than the new doctors, junior doctors coming in. Nothing wrong with junior doctors, but I think generally nurses are very, very good at doing sort of these sort of skills. And of course, they work close to the patient. So this is from the sort of manual that our nurses produced that is shared both at Gauche but also around the country. That's how they prepare, clean, and then eventually access. And then what happens, these patients have to sit there relatively still for 4 hours, and there are various tools that they can use, such as iPad, sometimes bribed with chocolate, And they can be quite active there, jumping up and down a bit too much. So, but on the whole, very few patients have to be sedated. Most of the patients are okay with getting used to the iPads and sometimes play therapy. So, as I said earlier, not all patients benefit in the same way. Some of them deteriorate a little bit from the start, and then maybe something in the long term. So, as I said, we worked with Wendy Haywood and the other team, the rest of the team of the biomarker discovery team, and we looked at the neurofilament light, which is a known biomarker of neuroinflammation in the CSF in this case. On the left, you see what happens to the NFL levels early on after the start of the administration of enzyme replacement therapy. These levels are huge. The normal level is certainly below 200 in the CSF. What you see is that different patients react slightly differently. On the right graph, what you see is that what happens a year or later after the administration. So even a year or year and a half later, some patients still have quite high levels of NFL, and only by about 2.5 years after the start of treatment, these patients' levels come down to close to normal or to normal. So different patients react differently. It may be that we will need to adjust the treatment somehow in order to be able to get these patients responding better. So next I'm going to talk about the treatment of the eye disease. So unfortunately, the enzyme replacement therapy that is delivered intracerebral ventricular, this enzyme does not cross the CSF eye barrier. I didn't know such existed before in most of these patients. So sadly, these patients lose sight. And that, of course, is a massive burden in terms of their quality of life and everything else for the families. So we work together with BDFA, the Batten Disease Family Association, and Isolated Families in order to try to do something. So we thought that the enzyme, the leftover of the enzyme at the bottom of the vial, we could use it and inject it in the eye, potentially, and maybe help to slow down at least the disease progression. So why did we decide that? So this is the work funded by BioMarin in the States. So on the top, this is the eye disease in the patients, where you see that the onset of eye disease is around the age of 4, and by the age of 8, these patients typically go blind. In the dog model, their onset of the disease is around 4 months, and by the age of about 9 months, the dogs go blind. So what these researchers did, they measured the retinal response in the dogs. What you see, the black dots, this is the normal dog with no disease, and essentially the ERG remains constant. The gray dots is in the dog that is deficient for TPP1, so it's a model of CLN2 disease, and you see the rapid decline in this retinal response. Whereas the dog that was exposed to the injections of the enzyme replacement therapy, after a few injections, the disease stabilized, and then their function remained. So what we did, once these papers in the dogs were published, we worked together with the ophthalmology team here at Gosh, and a lot of other, I have to say, sort of committees within the hospital. And Gosh is unique in having the ethics committee with the drugs and therapeutics committee and many other people to agree that we would do that, because this was a first in the world patient exposed to the enzyme replacement therapy in the eye. And also there was no cost to the drug because we just used the leftover at the bottom of the vial. So we worked with the families who raised money individually to pay for the 18-month program, which is now extended. And of course, there had to be a special coordination between the intracellular ventricular infusions and the injections in the eye, which were done and are done still in theatre. So could we make a difference? What you see on the left, this is a natural history of the retinal disease in the patients. In fact, this is the volume of the retina, central retina in the patients, and you see that to start with, it stays relatively flat. Then there is a period of rapid decline, and then there's a plateau where the retina is not declining any further. So what you see on the right, we did recruit patients at a fairly late stage in the disease, and you can see that all the patients on the right, because that's age in months, actually were already in this plateau stage of the disease where we didn't really make much of a difference. In the patients where there was still a rapid decline in the retinal volume with the arrows, you see that the orange dots is the eye where there was the injections. The blue dots is the eye which was the control eye, so the other eye. And you see that the orange, in at least the two patients, you see that the retinal volume remains the same after the first injections. So we hope that we're making some difference. This was a phase 1 study, so we've shown that this is a safe approach. We may need to change the dose, we may need to change the frequency of injections, but I hope we made a difference. So finally, I want to talk about the establishment of new centers. There was a delay of setup of the new centers because it happened seemed to coincide with the onset of COVID pandemic. So the MAA started in November 2019, and then we were supposed to transition patients to Manchester and other centers, and that of course didn't happen. So over the following years, the team here had 3 times the number of patients compared to what we were supposed to see. So I would say that that was pretty heroic effort particularly for the nurses who were there at the coal face, seeing these patients during these times. It was actually 6 centers, I can't even count till 6. So 5 in England and 1 in Northern Ireland that are now set up, and almost all patients that were supposed to be transitioned are now transitioned. And now there's more than 30 patients that are treated in these centers. So finally, I want to show you, so this is our laptop. So this is a patient that was treated from 20 months of age, and now she's 7.5. And I just wanted to remember that usually by 7.5, these patients populate over. So this is a child from Newcastle. She's arriving at Wash, very busy in the front. And here, 7.5. And then finally, she's come to the outpatient.
Wow! And then can you show me a few? What else did you do? Oh, did you do with your arms? We have you ripped out. Yeah, oh, that's amazing. Oh, it's going to be super impressive with you, Jessica.
Anyway, just acknowledgement, really thanks to many, many people. I can't really fit everyone on this slide. There are so many people that I have to thank.
Thank you, Paul, for a lovely talk. The videos really say it all, I think. Are there questions?
The delay between the onset of the condition and it being detected, so that 2-year gap between 3 and 5, if you had any inclination as to why there was that delay, and then I apologise if I missed it, but if you saw any differences in the children who received the medication earlier versus later, so is kind of giving that medication earlier better?
Yes. So the first question is why there is a delay in diagnosis. So the children usually start having seizures around the age of 3. At that point, they are usually seen by the general pediatrician. And the rule is that if these children are well and develop some seizures, then the first thing is to treat the seizures and not to further investigate. So by the time they are investigated, they are progressed significantly in that process. Whether that's changing, I don't know, maybe my neurology colleague could say, I hope that it's changing and the awareness of this is kind of pushing to earlier diagnosis. And also the fact that there is the treatment available, then that will push it first earlier. The second question, yes, definitely. I didn't have the time to show this difference, but yes, for sure. If you treat pre-symptomatically, she's like that at 7.5. Of course, I've shown you the best patient. If you start treating patient at the age of 5, so at the end of that video, actually, if I can show you, is her sister. Anyway, it's actually she's a lot more severe.
Stephen Martz from the CRF over here, Paul. Well done, and that was an excellent presentation. I just wanted to highlight the importance of PPIE, because wasn't it the patients and the parents who actually suggested initially that you can inject it directly into the brain, but our kids' vision are deteriorating. So actually it's important of involving patients in public in everything that we do in the BRC.
Yeah, absolutely. We have worked together with the families throughout, so yeah, absolutely.
Couple of questions from Slido, Paul. One, can you continue to have enzyme treatment for an unlimited period of time?
So this enzyme is on managed access agreement, which is a 5-year agreement between the BioMarin, NHS England and NICE. This period will finish on October 2024. So there will be a re-evaluation over the following year and then a new decision will be made.
You. And the second question, if starting treatment later is a problem, as you've discussed, is there a screening program being looked at in any way?
So ideally, I think that was brought up before, you would have neonatal screening or early screening. That would be an ideal situation. And I hope that when the newborn genomes come in, that CLN2 will be one of the part of the pilot. I think we're trying to develop, again with Wendy and her team, the biochemical method also to do the screening using blood spots. So yes, we're trying our best. Newborn screening is not an easy to get onto.
Thank you.
I'll ask you 2 questions. So should the paediatrician with a child with epilepsy just in the future just be able to do neurofilament as a test? Could that be, because that would pick up Battens and probably a lot of other neurodegenerative things, rather than worrying about doing the whole, this is treatable? And secondly, does your retinal improvement translate to visual function and functional improvement?
So the first one, the NFL, it's not specific. So whether that's helpful, I think that would need to be another study, how many disorders we would pick up. Clearly, it's not healthy to have high neurofilament light, but before you start offering it, I think you need to know what the interpretation and counseling is going to be. So I think that, it's an interesting idea. I haven't thought about that, but interesting idea to use it as a screening for a number of disorders. And then the second one was visual function is very difficult to analyze in children who have severe learning difficulties. So we're trying to work with the visual development team here and actually try to do that. It's not easy. It's quite difficult. So, but what we're hearing from the parents is that these children tend to look at things with the eye that was injected. Now that's subjective, so we hope we'll have some more objective measures of that.
There's one more question from Slido. It says, is there a way that you could tell if a child's eye has reached a plateau before starting injections?
So I don't know if you can from one assessment. I think it's quite difficult because even what I've shown you, the plateau was at different levels, so just measuring the volume is not necessarily going to tell you about the plateau. So not the one measurement, but more than one measurement perhaps, yes.
And finally, there's a comment on Slido that just says, well done team, great to see research translated for patient benefit.
Thank you.
Okay, so we have a break now until 3:25, and there's refreshments in the Winter Garden, and there's several charity partners here, so please visit their stands to find out more what they're doing, and we'll see you back here at 3:25. Thank you. So I think we'll make a start for the next session. So it's a great pleasure to invite Professor Christian Hedrick to come and talk to us. Christian's a Professor of Child Health at the University of Liverpool and Honorary Consultant Paediatric Rheumatologist at Alder Hey and part of our BRC's Paediatric Excellence Initiative. And we're really looking forward to hearing your talk on osteomyelitis and personalised treatments.
Yeah, thank you very much for the friendly introduction. Thanks very much for the opportunity to be part not only of the BRC, but of this really exciting meeting today. So I'll take a step back a little, and my talk's not going to be as exciting science fiction as the ones we've just heard. I'll talk about a project that's a bit behind on the translational pathway, and is now coming to an end when it comes to target identification. So our program is about molecular mechanisms in childhood inflammatory disease, and we nowadays stratify inflammatory diseases on a spectrum from so-called autoinflammatory diseases that are initially caused by dysregulations of the innate immune response, as opposed to autoimmune diseases that initially at the beginning of the pathophysiology are caused by dysregulation and pathological activation of adaptive immune responses. And today I'll talk about a number of autoinflammatory diseases, basically just mentioning that a number of monogenic so-called autoinflammatory diseases that all result in an increased activation of so-called inflammasomes and or uninhibited activation of IL-1 signaling can result in bone inflammation and have bone inflammation as a characteristic clinical feature. And then for the larger part of my talk, I'll talk about pathophysiologically complex autoinflammatory bone disease called chronic non-bacterial osteomyelitis, or CRMO, that we are focusing on in this project. Out of those single gene mutation caused autoinflammatory diseases with bone involvement, I just picked one, which is called Majeed syndrome. And in the past, this one has been referred to also as familial chronic non-bacterial osteomyelitis, or CNO. I would nowadays discourage from using this term and call it Majeed syndrome after the person who first described it. Majeed syndrome has initially been described as a classical clinical triad with early onset chronic recurrent multifocal osteomyelitis, usually with onset before the second or third birthday, in the presence of a congenital anemia and neutrophilic dermatosis. And initially, this condition has been proposed in 3 Arabic families, but we nowadays know that also families of European ancestry are present. So the molecular pathophysiology of Majeed syndrome is highly complex. We do know that it is caused by homozygous loss-of-function variants in a gene called lipin-2, and lipin-2 has been proposed to be involved in multiple mechanisms, including inhibition of MAP kinases. So loss of function in lipin-2, potentially in innate immune cells, results in increased activation of MAP kinases that can contribute to pro-inflammatory cytokine expression and release. And more recently, lipin-2 has been associated with cholesterol synthesis. And in a very interesting work, it has been shown that cholesterol is an inhibitor of a transmembrane protein called P2X7 receptor. P2X7 is an ion channel that allows the efflux of potassium out of the cells, and potassium efflux is a very potent activator of the NLRP3 inflammasome. And for those of you who may not know, inflammasomes result in the activation of inflammatory caspases, and caspases activate inactive forms of cytokines, including interleukin-1 beta and IL-18, that only after their activation get released from the cytoplasm and cause systemic inflammation. So this nicely explained why patients with Majeed syndrome have a pro-inflammatory phenotype that can be treated with IL-1 blocking agents, including anakinra and canakinumab, which resulted in clinical and laboratory remission in those patients. So as briefly mentioned, we are interested in a complex condition called chronic non-bacterial osteomyelitis, or CNO, an autoinflammatory bone disease that primarily affects children and young people. However, it can generally affect all age groups, including adults and also young children. However, that said, children younger than 3 years of age very rarely develop CNO. So in those patients, alternative autoinflammatory conditions that can come along with bone inflammation need to be very closely considered. CNO does span a broad clinical spectrum with sometimes mild monophasic and monofocal courses, but much more frequently, so that means in 85 to 90% of all cases, patients will have chronically active or chronically recurrent bone inflammation affecting long bones in a symmetrical fashion. However, that can also involve the vertebral spine, the mandible, or the clavicle, and that can result in significant pain, bony overgrowth, or even vertebral fractures in some of those patients. And in those cases, we talk about chronic recurrent multifocal osteomyelitis, or CRMO, which is sometimes used as a synonym to CNO. All that said, not all monofocal and all monophasic courses need to be mild. They can, especially when they affect the mandible or the clavicle, result in significantly pain and overgrowth, or when they affect the vertebral spine, result in fractures. There are associations with other inflammatory conditions, including skin disease, such as palmoplantar pustulosis and psoriasis, as well as severe acne, and some intestinal disorders, such as Crohn's disease or ulcerative colitis. And in adults, there is a symptom complex consisting of synovitis, acne, pustulosis, hyperostosis, and osteitis that is summarized as SAPHO syndrome, which we believe is very closely linked to pediatric CNO. And the reason for that is that all those individual features can also occur in children with CNO, however less frequently, and especially less frequently in their combination. So when we started our work more than 10 years ago, over the first couple of years, we showed that activation of toll-like receptors in monocytes from CNO patients with lipopolysaccharide resulted in an imbalanced activation of so-called MAP kinases. So while JNK and P38 MAP kinases were activated just normally as in healthy controls, MAP kinases ERK1 and ERK2 failed to be activated through phosphorylation, which resulted in reduced phosphorylation of a transcription factor called signaling protein, or SP1, that resulted in reduced nuclear shuttling of that transcription factor. And this transcription factor is very essentially involved in the expression of two immune regulatory cytokines, IL-10 and its homologue IL-19, that's located right next to IL-10 on chromosome 1. At the same time, MAP kinases ERK1 and ERK2 are involved in phosphorylation of histone 3 at position serine 10, which is an opening epigenetic mod that can open chromatin for transcription factor binding. So the combination of not enough transcription factor and closure of the promoter regions of those two genes resulted in reduced gene expression and a lack of those two anti-inflammatory or immune regulatory cytokines. And I was particularly interested also in the context of an animal model that was published around the same time, in which Greenhill and colleagues showed that IL-10 deficient animals in an induced arthritis model will develop increased bone inflammation and inflammatory bone loss as compared to wild-type animals. And they linked this to increased activation of the NLRP3 inflammasome, which again results in the activation of pro-inflammatory interleukin-1 beta and IL-18 that only then gets released from cells. And we saw something very similar in monocytes from CRMO patients, including increased activation of the NLRP3 inflammasome, resulting in increased release of pro-inflammatory IL-1 beta, And that was at least partially reversible by the addition of IL-10 and IL-19, suggesting that the lack of those two cytokines contributes to increased inflammasome assembly. Next, we looked at mRNA expression of IL-1 and inflammasome-associated genes, and indeed the expression of interleukin-1, NLRP3, the receptor molecule within the inflammasome, and an adapter molecule called ESC, which is encoded by the so-called PICARD gene, was increased in monocytes from CRMO patients on the mRNA, but also on the protein level. Esteban Ballester and his group in Barcelona had shown for patients with cryopyrin-associated periodic syndrome, namely Muckle-Wells syndrome, that reduced DNA methylation contributes to increased expression of inflammasome components and lastly to increased inflammasome assembly. And I should mention that cryopyrinopathies, including Muckle-Wells syndrome, are caused by gain-of-function mutation of the NLRP3 receptor, thereby resulting in increased inflammasome assembly and increased pro-inflammatory signaling. So we saw something very similar in monocytes from CRMO patients when we looked at those inflammasome-associated genes, including reduced DNA methylation of the NLRP3 gene and the PICARD gene that is encoding for the ESC adapter molecule. So Esteban showed that in Muckle-Wells syndrome, this was a result of ongoing inflammation, because when you treat patients with canakinumab and IL-1 blocker, this DNA methylation defect normalizes, and the DNA methylation patterns will normalize to what we see in healthy individuals. So we tried to do the same in monocytes from CNO patients, but we were only able to access a cohort of patients who received naproxen and reach clinical remission. So we will all agree that while naproxen can inhibit NLRP3 inflammasome activity, it is a significantly less effective treatment as compared to IL-1 blocking agents such as canakinumab. So we need to do a bit more of our homework there. And we did not see a normalization of DNA methylation pattern, which could mean that either naproxen isn't strong enough, or that this is a more upstream pathomechanism in CNO as compared to Muckle-Wells syndrome. So summarizing that, what we've shown to this point is that activation of toll-like receptors in monocytes from CNO patients results in a reduced phosphorylation and activation of MAP kinases, which contributes to the reduced expression of immune regulatory cytokines. That at least contributes to increased inflammasome assembly and pro-inflammatory signaling, which results in a distinct imbalance between pro- and anti-inflammatory cytokines. And from the world of endocrinology, we know that this balance between pro- and anti-inflammatory cytokines very centrally contributes to controlling the interactions between rank and rank ligand on osteoclast precursor cells, thereby controlling the differentiation and activation of osteoclasts. And therefore, we think that these mechanisms are very centrally involved in bone inflammation and inflammatory bone loss in CNO. So now I'll come to a number of unpublished data sets that are really interesting and we are working on as part of the BRC. So together with our colleagues in Iowa City around Polly Ferguson, we sequenced a number of interesting CNO patients, including a family with CNO cases over 2 generations. And what we did while using whole exome sequencing, we identified in silico predicted gain-of-function mutations in the P2X7 receptor encoding gene P2X7R. You will remember that gene from when I talked about Majeed syndrome. So P2X7 is a transmembrane ion channel that allows potassium efflux, which is an activator of the NLRP3 inflammasome, contributing in the activation of pro-inflammatory cytokines through caspase 1, but also the induction of pyroptosis, which is an inflammatory form of cell death, terminating inflammation. So what we saw in the mother and the index patient, both experiencing CRMO, when using a whole blood stimulation assay that was available back in the day when I was still in Dresden, was increased inflammasome assembly and release of IL-1 beta and IL-18 when compared to here 30 healthy matched controls. The release of interleukin-1 beta was reversible, at least in part, through inhibition of NLRP3 with a small molecule called MCC950, but when compared to healthy controls, this was a less strong effect. Next, we looked at the assembly of NLRP3 inflammasomes, so-called S-specs, which we can stain in live cells. And I don't know if you can see the little red dots here. These are assembled NLRP3 inflammasomes. These are monocytes from the patient as compared to a healthy control, and you can see here that over time, the number of assembled inflammasomes increased in the patient when compared to matched controls, very strongly suggesting that inflammasome assembly plays a distinct role in those individuals. So now to test whether this gene is important for a number of CNO patients or only for that one random family, we assembled a large national cohort of 191 patients from Germany and performed target sequencing of the P2X7 gene and identified 70 CNO-associated genes. So the data are relatively complex to understand, but overall we found a dramatically higher number of rare mutations in P2X7 in the CNO cohort as compared to a 1,873 bone healthy individual containing healthy control group, which only had 20 of those mutations. 12 CNO patients had rare high-impact variants, 11 of which were predicted to cause gain of function. One of them was proposed to cause loss of function. That very strongly suggests that P2X7 plays a pronounced role in CNO patients. To functionally test some of those variants, we generated genetically modified cells, so we deleted P2X7 or one or both alleles and then reconstituted with wild-type P2X7 and one of three variants we found. The index case gain-of-function variant, the one loss-of-function variant we found, and then one common gain-of-function variant that we also found in healthy controls, and then tested those individual steps that are induced by P2X7 activation. So what you can see here is potassium flux. So it's all based on what we see in the wild-type P2X7 carrying cells. Not surprisingly, the index case gain-of-function mutation increased potassium flux. The loss-of-function mutation reduced potassium flux, and the common predicted to be gain-of-function variant was pretty much the same as the healthy control. Looking at cytotoxicity, using LDH release assays, not very surprisingly, what we saw in loss-of-function mutations was reduced cell death. But very interestingly, after 2 and 4 hours, we also saw reduced cell death in cells carrying gain-of-function mutations. Looking at cytokine release, again, not very surprisingly, the loss-of-function variant had reduced release of IL-1 and IL-18, while the gain-of-function mutations had pretty much the same IL-1 release, but increased IL-18 release when compared to healthy controls. And that's particularly interesting in the context of reduced cell death, because those cells will continue producing and releasing those cytokines, and they can be re-stimulated while wild-type cells die. So taking all that together, altered activation of MAP kinases contribute to imbalanced cytokine expression in CRMO patients. Increased expression of NLRP3 and associated genes nicely align with reduced DNA methylation, which can explain increased gene expression. Whether that is an upstream event or the result of ongoing inflammation is currently unknown, and we're hopefully about to answer this question. Recent, currently unpublished data link mutations in P2X7 with CNO. So we find rare high-impact gain and one loss-of-function mutation in about 6% of the patients, and interestingly, they also have a different phenotype with more signs of systemic inflammation as compared to the remaining cohort. And overall, we find more rare variants, also of lower predicted impact, that could represent a risk allele for CNO. And that very strongly argues for cytokine blocking agents or inhibition of NLRP3 inflammasomes in patients with CNO. So this is summarizing what we currently have at hand to treat CNO. So we are already using anti-inflammatory treatments. So patients with CNO are frequently treated with TNF inhibitors if available. Currently in the NHS this is not available, but can nicely restore, at least in part, that imbalance between pro and anti-inflammatory cytokines. What we can access here in the UK is permethrinate. Permethrinate inhibits osteoclasts and also at least partially reconstitutes that imbalance between pro and anti-inflammatory cytokines in a yet to be determined manner. Naproxen is usually used as first-line treatment. Naproxen has very weak effects on inflammasomes. It can inhibit inflammasomes, but also reduces prostaglandin levels, which thereby inhibits the activity of osteoclasts. But based on what I've been telling you, there are additional targets. One of them is the inflammasome, and then particularly interleukin-1, and we're currently discussing with industry to move towards clinical trials, either blocking interleukin-1 or inflammasome assembly. And I didn't talk a whole lot about SAPHO syndrome, which is, as we believe nowadays, the adult counterpart to CNO. And one of the mechanisms that may play a role here and shift the phenotype from a mostly autoinflammatory disease to a more autoimmune phenotype that includes additional organ system involvement is the differentiation and activation of effector T cell populations through the chronic presence of those pro-inflammatory cytokines. And that could bring IL-17 and effector Th17 cells into the game, which may be addressable by, for example, IL-17 blocking agents or JAK kinase inhibitors that may prevent the activation or differentiation of those effector T cell populations. Thank you very much.
Thank you, Christy, for a very lovely talk. Are there questions? So do you think that it would be a combination of therapies that's required for CNO rather than a single? I mean, you showed you a nice slide at the end with a combinative kind of approach. Do you think that's the way forward for this group of conditions?
So we have to find biomarkers, I think, that don't only pick up differences between individuals, but also differences between different disease stages, and that's one of our targets right now. So we have a number of projects also looking in the serum proteome, which we will then align with age groups and different clinical phenotypes. So we do already know that some patients don't respond to permethrin, but do respond better to TNF inhibitors. There seem to be a couple of clinical predictors, including like female gender, high number of bone lesions, high CRP, but these are all like studies in retrospective large cohorts, which is the best we have right now, but not good enough, so we'll need to get better. And very few patients need a combination of the two, permethrin plus TNF inhibitor, and even fewer don't respond to any. In some of those, IL-1 blocking strategies have been used, so that is another strong argument for the use of those strategies in CNO, maybe already at an early stage to prevent more severe phenotypes from even happening. So yeah, we need to do more homework, but yes, there seems to be more than just one treatment for them.
Are there any other questions? Okay, so I'll close this session. Big thank you to our speakers and hand over to Dr. Sabir.